Students, in the prior lectures, we had studied about the fluid mosaic model, the structure of which explains the structure of plasma membrane, and I explained that it consists of lipids and protein molecules. The lipids are having polar heads and non-polar tails made up of fatty acids, and whereas proteins, they also have non-polar amino acids which forms their non-polar domain, and also they have polar domains which are formed by amino acids which are polar in nature and which are usually hydrophilic in their characteristics. So, fluid mosaic model, which was given by Singer and Nicholson, was the best and perfect model. And today, the plasma membrane structure is studied through this type of structure only. Unit membrane concept given by Robertson is no more considered. And I had explained some membrane proteins, which there are two kinds: integral membrane proteins and extrinsic proteins. Integral membrane proteins, they remain embedded in the lipid bilayer, whereas extrinsic membrane proteins, they lay outside the over the surface of the plasma membrane. The examples of integral proteins are porins, caderins, integrins, selectins, band 3 proteins, and glycophorins. Also, the very important antigenic determinants are present as integral membrane proteins. Then we have peripheral membrane proteins like spectrins, actin, and chirins. Then we had studied some membrane lipids which were phospholipids, glycolipids, and sterols. Then we had, after studying the structure, we discussed some of the functions of the plasma membrane. The first was osmosis. Because the plasma membrane is selectively and differentially permeable, osmosis is a very important phenomenon and is associated with movement of water inside and outside the plasma membrane, or extracellular fluid to intracellular and intracellular fluid to extracellular fluid, depending upon the concentration difference of solute inside and outside. Then the second function we had studied was diffusion. Diffusion is nothing but the movement of molecules from higher concentration towards lower concentration, and this depends upon the concentration. Then this was simple diffusion, and simple diffusion do not use any kind of carrier molecules. Another kind was facilitated diffusion, which is a very important form of diffusion. It employs a specific carrier protein called as permease to transfer the solute molecules, and it is faster and speedier than the simple diffusion, but the speed never exceeds the rate of change of conformation of the permease protein. This you can cite in this diagram, that after reaching a certain point, the velocity Vmax doesn't rise further, rather becomes constant. So this was diffusion. Then furthering, we had studied the mechanism of diffusion and how the solute and permease complex forms and it changes the conformation of permease. Then we had studied active transport, primary active transport and secondary active transport. In active transport, as I had mentioned, sodium potassium pump is involved in active transport, and in active transport, the difference between permease of facilitated diffusion and this active transport is that the protein sodium potassium pump or protein involved in active transport has ATPase activity also. So this was the mechanism, that 3 sodiums, they are thrown outside from the ICF and 2 potassiums are thrown in by the action of sodium potassium ATPase carrier protein. Then certain other functions like cell recognition, as I told you in the beginning, that the glycocalyx, which is formed by glycoproteins, is involved in this. And then secretion is the function of important function of plasma membrane. Oxidative phosphorylation in certain bacterias. Then endocytosis and exocytosis. Again, endocytosis is phagocytosis and pinocytosis. Then phagocytosis is cell eating and pinocytosis is cell drinking. If the substance which is taken in is solid, then it is called as phagocytosis, and if it is liquid, then it is pinocytosis. Then we had studied exocytosis. Chemoreception is another important function of plasma membrane, because receptor molecules, they are present on the surface of the plasma membrane. Plasma membrane or of the axon, which is neuron, part of neuron, is involved in conduction of nerve impulse. Today, students, we are going to study the cell junctions. Cell junctions are actually cell-to-cell interconnections. At the beginning of evolution, prokaryotes and protozoans were formed. Then protozoans were the single cells, and they started aggregating. But the aggregation of cells doesn't designate them as tissues. For the aggregation of cell to be called as tissue, there must be cell-to-cell interconnections, and they are very important for the integration of cellular activity and to carry out a particular function. The cell junctions, they also give tissues an architecture and a strength which is required. Otherwise, all of the cells included in the tissue will scattered with minimal of the disturbance. The first kind of cell junction is called as tight junctions, also called as Journali occludens. They prevent the lateral passage of the fluid, and they somehow seals the cells and prevents the lateral passage of the fluid through the cells in a tissue. They are composed of proteins and lipids. How do we come to know that they are made up of proteins? Because when they are subjected to enzyme protease and trypsin, they got digested. And owing to these tight junctions, the plasma membrane adjacent to the cells appear to touch or even fuse with each other. These tight junctions, they are present in the lumen of the intestine. They are present between epithelial cells. Nutrients are absorbed from the lumen of the intestines by cells, but taken in and released on other side into the blood by cell themselves, because the tight junctions, they do not allow to pass through these nutrients by the sides of the cells. Then the lumen of pancreatic acini, they also contains large number of tight junctions which prevents leakage of digestive enzymes into blood. Otherwise, the blood will be digested. You can cite the tight junction. The diagram depicts how the tight junctions they look. This is on the left and right side. You can see the cytoplasm of the cell 1 and cell 2, and then the solute molecules, they are trying to penetrate the lateral side, but you can see the tight junctions which are depicted in red color. They seals off the lateral passages. Then